Hey, hello everybody. My name is Matt O'Mara. I'm a professor at the Department of Computational Medicine and Bioinformatics at the University of Michigan. Our lab works on structural biology, but focusing on drug discovery, so structure-based drug design, and also analysis of high-content experiments. So I'll tell you a little bit about the University of Michigan, and then get into our department and then our lab. So Michigan Medicine is in Ann Arbor. We're in the Midwest, and we have a sort of a small town of about 100,000 people. We do have an enormous stadium that can hold about 100,000 people. And we are affiliated with University of Michigan campus, which has the medical school and also the undergraduates. It's an enormous university, and our department is situated inside of the Michigan Medicine, which includes the hospital and the basic science departments. To apply to our program, people typically come in through the program in the biomedical sciences. So this is an umbrella program that has around 500 faculty and 14 different programs that you could potentially join. So some of the basic science departments involved include things like biochemistry. We're in bioinformatics, which has its own department, and pharmacology and others. There are other schools on campus, including the College of Pharmacy, which has the Department of Medicinal Chemistry, as well as the undergrad MA, which has chemistry and biophysics and math and computer, and the engineering has the computer science. So there's a number of different schools. We're inside of the medical school. So to apply to the PIBS program, applications are due December 1st, so under a month. There's no GRE requirement, and our department, so when you apply, you specify which department you want to read your application. So we get around 200 applications to DCMB, and we make around 35 offers and about 18 matriculate. So that's a structure of one of the programs that you could come in through. Our department also offers direct admissions, but you have to join a lab right away and you don't do rotations. Or if you come in through the PIBS program, the program in the biomedical sciences, you would rotate through 3 different labs in your first year. Additionally, the College of Pharmacy and the medicinal chemistry department offers admissions, and that has a different set of requirements. Okay, our department is somewhat unique in that we have a full department of computational medicine and bioinformatics. This is from our recent retreat, and we have people who span doing genetics and electronic health records all the way through, we're doing structural biology, and others are looking at other analyses of metabolomics. So the program is often structured towards people who are trying to combine computational methods and applications in the biomedical sciences. We have people coming in through different tracks. So if they come in through more of a biology, biochemistry track, then they might take more CS and math classes. Or if they come in from computer science or math, they might take more of the biophysics, biochemistry side of courses. But the idea is that you have kind of the portfolio of different classes and expertise by the end. And then we also have affiliated with a larger consortium of labs that are involved in some molecular modeling or other types of computational analysis, and they might be on your thesis committee or prelim committees and things like that. Okay, I've been here at the University of Michigan for about 5 years. I've had my lab now for about a year and a half. This is our lab group from a recent lunch outing. And our lab is primarily focused on thinking about how we can combine different evidence and molecular modeling in ways of driving drug discovery. So we often use simulation-based models through, for instance, Rosetta, but also through statistics and deep learning-based approaches in order to anticipate how systems might respond to a perturbation. So the perturbation could be a drug or it could be a genetic modification, but we're thinking of it in terms of a dose response or a treatment effect estimation. So we're using causal inference and statistical modeling to think about how we can generalize to systems that we might care about, natural biological systems and others. Okay, so that's the overview of the slides that I wanted to share. And I think Camille is going to have some questions and we'll do a bit of a question and answer.
Thank you for your presentation, Dr. O'Mara. To start off, what is the focus and structure of the program in terms of coursework?
Yeah, so coming in trainees, depending on what type of courses you've had before, you might take either intro programming courses or higher level machine learning and biostats courses. But there is a set requirement of having genetic sequence analysis, data science, as well as topics that might range from metabolomics. We're offering a course in machine learning for drug discovery this winter. So there's a range of different sort of topic courses, but depending on the path, might take some of the foundation ones and then enough requirements to satisfy the degree.
Great. And can you describe the atmosphere in the department, for instance? How collaborative are people?
Yeah, I think we have a pretty good collaborative department. The physical space is partially located on Central Campus and partially on North Campus, which is a few miles away. But we have a good core group of labs that are in the department, and then labs that are affiliated in one way or another with it. We have a number of shared graduate students, so co-mentored with a couple of other labs in the program. So Yushwan here is with a neighboring lab that is working on single cell omics and trying to understand how deep learning works with that. So we're thinking about how treatment effect estimation might impact single cell resolution. But we do joint collaborative meetings, and we have I would say we're a pretty congenial group of people in the department. I think one nice thing about University of Michigan that might be different than, say, like East Coast or West Coast environments is that because University of Michigan is so huge and there's not a lot of other big institutions, say in Boston or New York or San Francisco, that there's a real collaborative nature in terms of being able to approach almost anybody who's around, and there's good opportunities for infrastructure support and doing larger collaborative projects all within the University of Michigan. So the friction of trying to get projects off the ground is very low.
Great. Does the program have lab rotations? And if so, how many? And what are they like?
Yeah, so the typical rotations through the PIBS program are you do three of them, and they're 8-week rotations. And the idea is that you would pick out labs that you might be potentially interested in joining. And then through those, you come into the environment, explore some new biology or new bioinformatics, and then see if it's a good mentoring environment. And then at the end of the rotations, you would join. It's possible to do a summer rotation, but it's not required. But the idea is that you have to do at least 3 of them before you can pick a lab to join.
Okay. What are the typical career paths for students coming out of the program?
Yeah, a lot of people end up going on to doing more research. So either as postdocs or in industry. We have a good network of people coming out of our department in different industry positions. And a lot of them take the bioinformatics skills or the data science into a range of different areas. But there's an incredible need for strong biotechnology and expertise. And so I think there's a strong support for doing research in one form or another. And I would say probably it's split 50-50 into academic and industry.
Thank you. What are the application requirements and what is the application deadline?
Yeah, I think our application requirements are similar to other institutions. So we don't require GRE, but we do have 2 statements. So one is a research-oriented statement and one is a personal statement. And then we ask for a CV and then letters of recommendation. So we get all these packets for each applicant, and then the part of the admissions committee will process them. And then based on the ranking and scores and feedback, we'll invite people to interview. And we have usually 2 on-site interview weekends in January and February. And then from that, we'll make offers, and then hopefully they'll accept.
Thank you for that information. And how competitive is admission to the program?
Yeah, we tend to get a wide range of people, international and national, applying, and we generally have a very strong pool of applicants. So I mentioned earlier, we get around 200 or so applications to our department, and we end up making offers to around 30 of them. So that's no less than 20% acceptance rate. But typically what we're looking for is really good fit for the program. So that means a really strong academic background, a research experience is definitely a plus. And then also having a good idea of what you would want to get out of the program and how you would do that. So maybe a couple of faculty that you think you would be interested in working with or at least rotating with, and then how you'd benefit from the specific training opportunities that are in our department and also at University of Michigan. And from this, we would evaluate the applications and try to make an assessment of it. We typically don't prioritize domestic applications, although some other programs tend to prioritize domestic applications. So we definitely are very internationally friendly in terms of recruitment.
Great. And lastly, where can I find more information about the program and the contact information?
Yeah, so our department is the Department of Computational Medicine and Bioinformatics. So DCMB, and then or University of Michigan. There's, I think, probably a Google search away to find it. But if you're not finding the information that you particularly want, or you want to know more details, I'd be happy to set up a time to chat with anybody from the Rosetta Commons to come and learn more about our program and what opportunities might be available in our lab or other labs in the program.
Great. Thank you so much for joining us today, Dr. O'Meara.
Okay, great. Thank you so much for the questions and good luck.
Okay, hello. My name is Colin Smith, and I'm a professor in the chemistry department at Wesleyan University. And I'm also associated with the molecular biology and biochemistry department as well. And so I accept students from both departments. And the PhD program at Wesleyan is unique in that we're primarily undergraduate university, but we have a strong PhD program. And one of the advantages of that is that we do have a little bit smaller labs, and so you get a lot more personal attention. The chemistry and MB&B departments are each about 10 faculty. And so you basically will, coming to Wesleyan, get to know every single faculty member in the department, and they'll all know you, and you'll know all the students. And so I think it's a really nice environment in that way. And so we do have a really supportive and diverse learning environment. And so we do all sorts of different research from biophysical to biochemical, also synthetic, polymer, computational, et cetera. And so I'll just tell you a little bit about the kind of research that we do in my lab. And my lab is really interested in trying to redesign protein energy landscapes, particularly in de novo computationally designed proteins. And so we work with and try to understand the biophysics of de novo designed proteins and how that influences their function. And we also do look at some naturally occurring proteins, although my lab's focus is increasingly on de novo designed proteins. And so we do that with a combination of computer simulation, protein design applications, and also NMR spectroscopy. And as part of that, we develop new computational methods for simulating proteins. And one of the things, though, with these simulation methods is they're not always accurate, but to really make the most out of those simulations, we try to fuse the simulations with NMR data. And we also then, when it comes to NMR data, we develop new methods for actually pulling the raw data out of the NMR spectra. And that's a big focus of the lab as well. And so I can just say a little bit about the primary application areas we're working in. We are working on understanding the dynamics of a new class of de novo designed proteins and understanding how the dynamics of this small molecule ligand then affect the function of the protein. And we think ultimately this research could have a lot of downstream applications in terms of understanding how a protein stabilizes a particular conformation of a ligand for things like enzyme design. And then the other area we're really pushing into now is trying to optimize the dynamics of de novo designed mini proteins that can be designed to bind to and inhibit the surfaces of target proteins. For instance, the SARS-CoV-2 spike protein. The computational designers in David Baker's lab were able to design a binder within 5 months after getting the structure. But with both of these kind of design application areas, we really don't understand after the design how well do those designs actually fit the target structure or how flexible are they. So we're really at the cutting edge in terms of trying to understand that. And so I just wanted to give one other little bit of brief background in that we're going to be moving into a new building in the next year or two. So we're target to moving in the 2026. And so that new building is going to house the chemistry, molecular biology, and biochemistry and the biology department. And so it's going to be great brand new facilities. And so that would be the kinds of things you would get to experience and take advantage of if you came to Wesleyan.
Thank you very much, Dr. Smith. Can you describe the atmosphere in your department and how collaborative people are?
Yeah, the atmosphere is very collegial. I think we try to be supportive of individuals and especially any needs they have, and we are pretty collaborative. We have a strong focus on the molecular biophysics, which I really appreciate here. That kind of spans both the chemistry and MBMB departments, and so I talk to and work with many of the faculty in each of those departments.
What is the focus and structure of the program in terms of coursework?
Yeah, the chemistry department, if you came there, then there's 5 core courses in biochemistry, inorganic chemistry, physical chemistry, quantum chemistry, and organic chemistry. And so those would be courses that you can actually get out of if you can display sufficient previous knowledge. In the MBMB department, there's a little bit more flexibility, and it's again trying to strengthen your background in both molecular biology and biochemistry slash biophysics.
All right. And does the program have lab rotations? And if so, how many?
Yeah, so the chemistry department has 3 lab rotations that you'll do in the fall. And so those are typically about a month each. And then the MBMB department has 2 lab rotations that are each about a semester long. And so that's a little bit more of a extensive experience in a lab.
All right. And what are typical career paths for students coming out of the program?
Yeah, so I would say that many of our students, I'd say most, go on to postdocs. And then after postdocs, probably the majority of our students go into industry, although we have had many faculty also go on, or many students go on to faculty jobs. We have a former alumnus who is at Harvard, for instance, right now.
And can you describe the application requirements, process, and deadline?
Yeah, so the application requirements are we don't require degrees. We basically are looking for 3 letters of recommendation, a personal statement, a research statement, a CV, and that's about it. And the deadlines are a little bit rolling, but for best consideration, you should have your application in by December 15th.
Okay. And are there interviews required in the process?
That's done on a case by case basis, depending on the department. Sometimes there's interviews, sometimes we do direct admits.
Okay. And how competitive is admission?
Yeah, at least in the chemistry department, we typically get about 70 applications each year, and then we'll admit anywhere from 5 to 10 students.
And lastly, where can I find more information about the program and contact information?
Yeah, Google is your friend, but if you want to type in a URL, it would just be wesleyan.edu/chem for the chemistry department, or for the MBMB program, that would be wesleyan.edu/MBB.
Hey, thank you so much for joining us today, Dr. Smith.
You're welcome.
Hello, everybody. I'm Scott Horowitz. I'm an associate professor at the University of Denver. I'm in the departments of chemistry and biochemistry, and also the biophysics program. I'm also a principal investigator in the Nobel Institute for Healthy Aging. And yes, if you're curious, looking at this photo, I took this out of my office window, so you can see the whole Front Range from DU's campus. So DU is a small research university in terms of number of people, but we put out a ton of great research. So on a per person basis, we stack up with just about anybody. And in my opinion, that's a great training environment for students, because you get to have a lot of personal interaction in the kind of smaller numbers and smaller space, but still have the opportunity to put out tons of great research and learn a huge amount. So about my lab. So my lab does a couple things. The primary thing we work on is how RNA and nucleic acids affect protein folding. So I'm guessing most of you about protein folding, basic thing that Rosetta Commons works on, for example, on how proteins need to fold to be functional, and that when they misfold and they aggregate, that can cause a lot of really nasty diseases. What probably you don't know as much about is that nucleic acids, especially RNA, interact with proteins during all of these different processes and affect how proteins fold and misfold, and can dictate, for example, diseases like many different neurodegenerative conditions. And so in my lab, what we've done is we've worked on figuring out which RNAs are responsible for this. And regular biology is also disease. We've determined how RNAs affect protein folding and protein misfolding, what roles they play in disease. But now we're starting to even think about how our protein folding systems that we have in cells even evolved in the first place. and how we moved from RNAs doing this to eventually having proteins to help with protein chaperoning and folding type of problems. And we go about this through a combination of techniques, so a lot of biochemistry, biophysics, genetics, structural biology, to get at all these questions. And we do things that range from basic biology all the way to medical applications. So this is what my lab primarily works on. We also work on the computer game Fold It. So I co-run Fold It. It is a biochemistry video game where players compete and collaborate on a variety of different biochemistry puzzles that are real-world puzzles. What my lab does with Fold It is two things. We work both to incorporate experimental data into the game, especially from crystallography and cryo-EM, and we also develop educational tools for Foldit. And so our educational tools that we've developed are used by classes worldwide. And that's the summary of what we do at the University of Denver. And I'm happy to take questions and hope to get more questions later after people watch this.
Thank you very much, Dr. Horowitz. My first question is, can you describe the atmosphere in the department and how collaborative people are?
Yeah, DU is small, as I mentioned, and so we see a lot of each other. The buildings are all right next to each other. I see all of my colleagues and most of the students from the other labs on a really regular basis. And so that creates for a good collegial atmosphere. People get along quite well. Collaboration, there's lots of collaboration between the labs. Obviously, that's driven by people having interactions with each other, being interested in each other's research. And I think that's something that we typically do pretty well here at DU, is that because we see each other regularly and we talk to each other in pretty good depth, collaborations form pretty easily.
And what is the focus and structure of the program in terms of coursework?
The two programs I'm involved in, so chemistry and biophysics, have different structures. So chemistry is a little bit more traditional in that it's a little bit more coursework heavy in the first year, and then you get into the research more in your second year. Biophysics gets you a bit faster into the lab, I think, and you have a few more options of people who are not just in one department, but you have a wider variety of people to choose from. You don't need to apply to them separately. You can actually submit one application and choose your program if you're accepted to both, for example. So it's a situation that I think works out well for students where, depending on what it is you're looking for, you have options.
Great. And what does the program's lab rotations look like?
Yeah, so this again is slightly different in chemistry and biophysics. So in chemistry, we have one required rotation and then you can do more, so up to three. And those rotations happen depending on when you start. So they could start over the summer, they could start in winter, they could start during the quarter. There's a fair bit of flexibility with it. Biophysics is a bit more rigid in that you do 2 to 3 rotations during the quarter with regimented times of start and end. So it's slightly different systems for how the rotations work, but like most places, you go and you talk to different professors whose work you're interested in, and then you kind of come up with which ones are most going to be best for you, think, and then it's just a matter of figuring out whether you're rotating in a lab that everybody else is rotating with at the same time and just trying to sort that out. And the faculty help you sort that out too. And then you do your rotations and you take it from there.
And what are the typical career paths for students graduating from the program?
So we have a variety, obviously. So a lot of our students go into industry, straight into pharma or chemical company type industry. We have a number that go to postdoc. We have some that go to government. We have some that go to teaching. So it's quite a variety. Denver being a relatively large city and with Boulder and such right nearby, there's quite a number of chemistry and biophysics type of companies in the area as well. So it's not uncommon to have people graduate from our program and to actually be able to find a job locally.
Great. And can you describe the application materials and process?
Yeah, it's pretty standard. There's a general Office of Graduate Education webpage for DU where you go and you find all of the graduate programs applications. From what I recall, it's pretty standard personal statement transcripts. We don't require GRE, letters of recommendation, and CV are the other materials, I think. And again, you don't need to apply separately for biophysics and chemistry. You can just apply for one and then just select, please have this other program also review my materials.
Great. And what is the application deadline?
Oh boy, I'd have to check. I think it's in December, if I recall. But the one thing that I will mention is that we again, we're a slightly smaller school, slightly smaller program. We're willing to work with students on the deadline if somebody is a little late. But that's that we've taken students who have turned in their applications late before, sometimes quite late if they're really a good student, because we're looking to get the best students, and this is a long process anyway. And if you're just not going to make it quite on time, shoot me an email, and I can let the people who are looking for the applications know, hey, I think we can get a really good application from this student. It's not here yet, and we could potentially take it late even after the listed deadline.
Oh, wow. Okay. And how competitive is admission?
Good question. I'm not on the admissions committee, so I don't know exactly how competitive it is. I
Mhm.
would say we definitely turn down students every year, and we definitely take students every year. We usually have an incoming class of about 8 to 12 chemistry students and about 5 to 7 biophysics students. So there's definitely competition, but I don't think that it's a school where if you're like, oh my God, I don't have a 4 point, or oh my God, I didn't get a publication out of my undergrad work, apply, right? We have lots of students come in who have gaps or pockmarks, but we're looking for people who love science and have shown that and want to keep doing that.
That's awesome. Lastly, where can I find more information about the program and your points of contact?
Yep, du.edu for general DU stuff. The chemistry department and the biophysics program are pretty easy to find.
Mhm.
Either from that page or a quick search away. There you can find descriptions of all the labs and what they do. You can find lots of information about and all of that. If you have questions, please email me. I'm also not hard to find. My email is scott.horowitz@du.edu, and I'd love to answer questions from people who are watching this.
Great. Thank you so much for your time, Dr. Horowitz. We appreciate it.
Yeah, thank you for the opportunity.
Thank you so much. It's my great pleasure to give an introduction about what we have been doing to all the students. My name is Zhang Yan. I'm an assistant professor from Vanderbilt. So when I got started, I see a big market in protein engineering. That's the cornerstone of modern chemical engineering. A super big market, but behind the idea is a very simple principle, which is what you get what you screen for. So when I started around 4 years ago, I had a super naive question, which is how about we get what we ask for? That's the reason why I'm thinking maybe the future scientists, the way how they do protein engineering will be similar to how we use Amazon Alexa these days for music selection. So if you need a mutant for therapeutics, for enzyme catalysis, you can just talk to the computer like the way we use Amazon Alexa. So I call it Alexa for mutants. That's where the name Mutexa come from. So behind Mutexa is actually a very big program in terms of doing a high throughput protein modeling. And I have a big team of students working on those led by QZ Xiao and Chris Jurish. And the core idea using Mutexa, we're trying to do what we can now do using traditional enzyme engineering strategies. One of them would be code adaptation. A lot of people are focusing on how to make proteins more stable under thermal conditions, but code adaptation is more like the other way around. So the core idea is that if you decrease the temperature by 50 Celsius degree, how much can you still make the enzyme remain active? And it turns out that using Mutexa, we're able to create enzyme, find design principles that make enzymes still remain very active, even at 0 Celsius degree. So if you want to know more, feel free to scan the QR code. And the second part of the research, still by using Mutexa, we go beyond enzyme and look into small peptide like lasopeptide. And this is a part of research that we're very excited about because a lot of those lasopeptide occupied around 20 to 30% of natural product drugs that are used that are approved by FDA. And the structural prediction of natural product peptide, especially ribosomally synthesized post-translationally modified peptide, is a big challenge that has no solution. And by leveraging the Mutexa system, we're able to get a structure and also get the structures predicted by using AI combined with physics. And for here, I just want to give you a quick demonstration about a computational web interface we have developed. So from here, you can get a database, and from where you can get the lasopeptide structure. Many of them are potential antimicrobial drugs. And you can also go to the interface by if you have a specific sequence where you want to get a structure, and you can also use our interface by inputting the lasso peptide sequence and get a target name. And of course, you need to provide an email so that we can give you any update on what's going on with the lasso pred, and from there you can get a structure predicted within 5 minutes. So we're excited about this possibility because being able to get natural product structure predicted is something that AlphaFold can now do properly. And we also see great opportunities to expand computational prediction by integrating multiple tools in molecular mechanics and AI. So I just want to quickly thank my entire group and thank Rosetta community, NIH, and Vanderbilt University for the funding support. I'm happy to answer any questions.
Thank you so much for your presentation, Dr. Yang. To start off, can you describe the atmosphere in your department and how collaborative people are?
Thank you. So the department is very collaborative because we have a super close spatial proximity between our medical center and Vanderbilt Department of Chemistry. So many of our colleagues are talking to each other, strategizing ideas. And for me, I had an ongoing collaboration with Professor Jens Meiler, who is an important person from Rosetta community, and also Brian Buckman, a natural product chemical biologist in our department. So we always collaborate with each other. The department is pretty small, so it's also very supportive for student growth.
And what is the focus and structure of the program in terms of coursework?
So it depends on which direction the student want to go. For me, I specifically recruit a student from the physical chemistry division. That sense, student will complete quantum mechanics, statistical mechanics. Of course, they are free to select. There are many elective courses, including computational modeling methods, analytical chemistry, and also computer-aided drug design. So in general, the coursework is not super intense, but also abundant enough so the students can actually get a lot of knowledge in different aspects centering around computational biology and chemistry.
And does the program have lab rotations? And if so, how many?
So there are 3 lab rotations starting from fall. So the goal for the 3 rotations, students are free to choose different labs and they can get immersed, maybe get involved in a small project. And for students who are very determined to join one single lab, it's also possible to negotiate for getting this component removed. But in general, I highly recommend the student to try different labs because that's also serving as an opportunity to nucleate potential collaboration or finding co-mentorship.
What are the typical career paths for students coming out of the program?
The majority of the students from the department are pursuing a industrial position right after graduation. There are Pfizer, Merck, and many other big pharma companies, definitely one of the destination for, one of the career path for students to pursue. But a substantial number, like 20% of our graduates also go to different top institute for postdoc position, including MIT, Harvard, Princeton. And we definitely have, and after the postdoc, a lot of students actually land in academic positions, including either R1 Institute or PUI. So the career path is definitely diverse.
Awesome. And can you tell us about the application requirements and process?
Sure, the application requirement is pretty standard. We require students to have CV, research statement, 3 letter of recommendation, transcript, and also we don't require GRE anymore. And for international students, TOEFL or Duolingo are also accepted. So it's very standard, similar to other schools. Just to clarify, are you also asking about the procedure for application?
I just wanted to know the materials that they had to submit, as well as once they've applied, when they can expect to hear about interviews and the deadline for application.
So after they applied, it probably take 1 to 2 months for them to hear about the interview. We don't necessarily have an interview, but for some students, if we want to know more about what the student have been doing, their specific interests, we may offer to talk with the student, especially this will also enhance a mutual understanding between the student and the faculty members they want to work with.
And how competitive is admission?
So the application process, we typically receive 200 to 300 applications, and we will finally have around 20 students.
Great. And I do see that you have shared your contact information here as well as the website for admission. So thank you for that.
Sure.
Thank you so much for joining us today, Dr. Yang. We really appreciate your time.
So there's one more thing I want to clarify. So
Sure.
the deadline for the application for the early submit, application deadline will be December 1st. I highly encourage students to apply before this time because that they will get fee waiver, so they don't have to pay the application fee if they are early enough.
That's great to know. Thank you so much.
Yeah, thank you so much. I really appreciate the opportunity.
Thank you for joining us.
Hi, I am Claire Woodward. I am not Nick Sgourakis. Nick couldn't be here today, so I am a fifth-year PhD candidate in his lab. So I'm going to talk today about my program and also a little bit about the lab as well. So I am in the biochemistry, biophysics, and chemical biology PhD program, or BBCB for short, at the University of Pennsylvania. Just very briefly about the program, we have a really strong interdisciplinary focus. I'm sure you can get that just from the name. Biochemistry, biophysics, and chemical biology are really three separate fields that we have mashed into one. So we have people in the program who study anything ranging from structural biology to straight up metabolomics, RNA biology, you name it, we pretty much have it covered. And there's a lot of collaboration within the department and within the grad group as well. So we have a huge number of students who collaborate with other labs within the program itself or on campus or more broadly at other institutions. The program also has a lot of focus on diversity and inclusivity. That is a very key tenet of our department and something that we have especially been focusing on in recent years in terms of recruitment. And within the program, there's a really strong sense of community amongst the graduate group cohorts. So a lot of times, especially the newer recruits will become very close friends with the other students in their cohort and do a lot of things together as they've all moved to campus and are getting used to the environment. So we have a lot of community building activities as well. And just a little bit about my lab. Like I said, I'm in Nick Sgourakis' lab at Penn. We're also affiliated with the Children's Hospital of Philadelphia. Our lab is physically in CHOP, but we're also in Penn. He's co-appointed, and they're very close in space. They're actually next to each other. So there's a lot of collaboration within the Children's Hospital and Penn Medicine as well. Just very briefly, generally our lab focuses on studying a protein called the major histocompatibility complex, or MHC. And MHC, I think I would like my pointer here, hang on. MHC is a really key component of the adaptive immune response. Proteins that are just in your cell, or if you have an infection, viral proteins that enter your cell. Also, in a lot of cases, cancer mutations create sort of new proteins in the eyes of the cell. Regardless, any protein that is there is going to be chopped up by the proteasome to create these peptides. And what happens is the cell then takes those peptides into the endoplasmic reticulum and uses a complex of proteins and chaperones called the peptide loading complex to load those peptides onto MHC molecules. Now, MHC is actually the most polymorphic gene in the human genome. So every single person statistically has their own set of MHC molecules that they use to present peptides, which means that which peptides you present and how high their affinity is for the MHC complex itself depends on the person, which is why some people are more susceptible to certain diseases than others. But regardless, when a high affinity complex is made between MHC and a peptide, it is then transported to the cell surface. And at the cell surface, it is surveilled by T cells. And if T cells identify a peptide that is foreign, for example, from a virus, it will trigger an adaptive immune response and kill the T cell. And so this is the process. We study all aspects of this process from beginning to end, including which peptides are presented on which MHCs and which chaperones are used to facilitate that process in the ER, and how that complex is transported to the membrane and how stable it is once it's at the membrane. That's the work that our lab does. And even going further than that, if you're familiar with CAR T cells, which are synthetic receptors on T cells that we engineer to use for cancer therapies, our lab has in recent years been also working on designing new T cell receptors that are specific to cancer-based peptides. That's a really key aspect of our lab recently as well. Technique-wise, we're very lucky at Penn to have access to a lot of really cool cutting-edge techniques. So we have our own NMR facility. We have a 600 and an 800 megahertz NMR system. Because our lab does a lot of NMR, we have almost unlimited access to this facility at any time. So we collect a lot of data just at will. We also have access to the Johnson Foundation Biophysics and Structural Biology Core, which has a cryo-EM as well as crystallography tools, although we also have our own crystallography robot in-house in our lab itself. We also have several really cutting-edge techniques like ITC and SPR right in our lab, which is a very important privilege that we have here in the Sgourakis lab. And we also, because we're also part of CHOP, we have access to their flow cytometry core facility, which is very extensive and allows us to take the biophysical studies and the engineering that we've done in the lab and convert that into functional studies on how T cells actually kill tumors. So that's a brief overview of kind of my lab and also the grad group itself. So here's some important contacts if you're interested in learning more about the lab. Here's our website, Nick's email and my email. Please reach out to me with any questions if you're interested in hearing more. I would love to talk to you more about that. We also have the BBCB, the grad group chairs information here, as well as our website. And just to say, the vice chair is actually Nick, so you can also reach out to Nick for more information about the program itself. And yeah, thank you so much for listening. And like I said, please feel free to reach out if you have any questions or want to hear more.
Great. Thank you so much for your presentation, Claire. I just have a few questions about the program.
Sure.
Can you describe the atmosphere in the department and how collaborative people are?
Yeah, it's a very collaborative program. Like I said earlier, it's very common for students or for PIs within the department to collaborate with each other, to collaborate with other departments on campus. It's actually so common for these collaborations to happen that actually a very large number of students in the program are co-mentored. So that's how close these collaborations tend to be. And we're all in very close walking distance to each other. I think the farthest lab away from me in the department is about a 10-minute walk from where I am right now. Everybody's here and everybody's working together and talking to each other all the time.
Great. And what is the focus and structure of the program in terms of coursework?
Yeah, so we do have some required coursework in the first year and the first half of the second year, so the first 3 semesters. It's really designed to get students up to speed because, like I said, the program is pretty interdisciplinary, so we don't necessarily expect everyone coming in the program to have the same academic background. We've had people come in who were majors in biology, and we've had other people come in who've only done strictly computational work or strictly physics or something of that nature. And so we have to get everyone up to speed with sort of the same baseline knowledge. So the coursework is really designed for that purpose. And it's pretty general knowledge based. There's not a lot of difficult exams. There's not a lot of heavy homework and a lot of hard work that you have to do outside of class. It's really just designed for the purpose of helping students learn and get up to speed before they join a lab.
Okay. And does the program have lab rotations? And if so, how many?
Yes, so we require 3 lab rotations. They will all be in the first year. Usually one of them is the fall semester, and then there's an early spring semester and a late spring to early summer 3rd rotation. They're usually around 8 to 12 weeks. There's some flexibility there. Students can choose when to start and end their rotation within a certain window. We typically expect that at least one of those rotations is with a primary faculty in BBCB. And I say that because we actually don't require any of our students to strictly rotate within the grad group. And in fact, you can join a lab that's not affiliated with the grad group. We just have to affiliate that PI with the grad group then, which is really just signing a paper, and it takes about 45 seconds. So it's very common for students to rotate from within the grad group, but also like more broadly, any lab at CHOP or Penn is an option. Those rotations are really just an opportunity for students to try out the lab, try out maybe new research that they haven't done before, meet the people in the lab, decide if the mentorship is right for them and if it's a good fit. We also don't require you to join a lab after 3 rotations if you decide, I didn't think any of those were the perfect fit for me. We've had students do extra rotations. We've had students do 2 extra rotations if they have to. And it's not a big deal. Those students don't fall behind or anything like that. That's also totally fine.
Great. What are the typical career paths for students coming out of the program?
Yeah, it could be really anything. We have a really strong career support network here at Penn. So as a 5th year, I'm now starting to see some of my friends move on to jobs. I'm also starting to think about that. We have students go on to do postdocs and go on the academic track. We have students go straight into industry. We have students do biotech. We have students do science writing, government, teaching, just across the board. And we have a network and support for any path that you think you might be interested in. We also encourage internships and such as you get toward the end of your PhD, if that's something you're interested in pursuing as well.
Okay, that's great. Can you share the application requirements, deadline, and process?
Yeah, so BBCB, it falls under the umbrella program of biomedical graduate studies, which is really just for the purpose of recruitment. Otherwise, we don't do anything within BGS really, except for they tell us how many students we can take per year effectively.
Mhm.
But you apply through a broader BGS application. So just make sure you check the BBCB box during that application. It is due on December 1st. It's pretty straightforward. It's a research statement, a personal statement. I think we also have a diversity statement in our program. We ask for a CV, we ask for letters of recommendation and a transcript. To be honest with you, the most important part of that whole application, though, is the personal statement.
Good to know. Do you know how competitive the program is for admission?
Yeah, so it does depend on the year. Like I said, BGS does dictate a little bit how many students we can take per year. So usually I believe we get around 150 to 200 applications per year in our specific program. We typically accept around 30 of those. But I will say the vast majority of people who are invited for an interview do get an acceptance. So we typically only invite for the interview the people who we're pretty sure about already, and that the interview is just to make sure they're a good fit with people and the environment here at Penn.
And I see that you have shared all the relevant contact information with us, so thank you for that. And Claire, thank you so much for your presentation and answering our questions. We really appreciate your time.
Yeah, thank you so much for having me. Again, sorry Nick couldn't be here, but I hope I've covered all the bases regardless.
Yes, definitely. Hi, everyone. My name is Camille Mathis, and I am the program manager for the Research and Mentoring for Postbaccalaureates program through Rosetta Commons. Our program is very unique in that it is a cyber-linked program. So we have various students placed in our host labs all over the country. Rosetta Commons has a network of over 100 labs. So our participating labs are, and they're all doing different science. So you are bound to find a lab and some science that you are interested in. The RAMP program for postbaccs is for students who have graduated within the past 4 to 5 years and are looking for additional research experience before they apply to a PhD program. The RAMP lasts for 1 year, and students are placed at our host labs after attending our boot camp training. That training lasts for a week, and it is done at Johns Hopkins University. And basically, students learn the community coding environment. They learn how to basically do our science so that they're prepared to go into one of our host labs. And they also cohort build and get to know one another. That's one of the great things about the RAMP program, is that you do have peers there with you going through the program, even if they're not in the lab with you. And now these cohorts of students do meet monthly to do journal clubs and chalk talks, and then there are also group activities. For instance, we are going to have a group trip at the end of the year. These students also present their research in the winter at our Winter Rosetta conference. So it's a great time to show what you have been working on, network with industry and PI professionals, and really immerse yourself in the Rosetta community. Our application for the RAMP program is open now. It's available on our website. And we are also hosting a RAMP information session on December 4th, and that registration is available on our website as well. And we hope to see anyone there who is interested in getting an extra year of research before they apply to a PhD program. We do also have quite a bit of PhD prep. We review your statements of interest as well as doing graduate school mock interviews, because we really want you to be successful and be prepared to enter graduate school after you are finished with our program. So we really look forward to seeing you on December 4th.